Hello and welcome to Nikolai's genetics lessons, and this video going to be about forensic science. As usual, I recommend you to pause video here, read the questions, try to choose the correct answers, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. So here is the first question. Probability calculations are used in forensic applications of DNA fingerprinting to determine if, and here is 5 answers to choose from. Answer A: DNA from 2 different sources have matching alleles. Answer B: DNA samples were degraded before analysis. Answer C: a match between alleles of different DNA samples might have occurred by chance. Answer D: an unknown DNA sample is from a male or female. And answer E: alleles in the suspect's DNA are different from alleles of victims' DNA. Of course, using DNA profiling, we can tell whether this is DNA of the male or female, if it is different from suspect's DNA or victim's DNA, and we can also answer such questions as if this DNA evidence is degraded or not, because if would be degraded we wouldn't be able to get results. Of course, we can work with very degraded samples of biological samples, but also to a certain degree. Sometimes we have to use, instead of nucleus DNA, only DNA of the mitochondria, because mitochondria DNA present in much higher copies, and it is much smaller, and also can be found in such tissues that is not very easily degradable, like hairs, nails, and bones. So actually you have to pay attention in this question to word probability, and forensic scientists have to find probabilities that some evidence from the crime scene match that of the suspect, I mean DNA profile, in order to show in the court, to show the judge that coincidence of occurrence randomly of two such profiles would be very negligible number. For example, normally we are saying about 1 in tens of millions or 1 in tens of billions. Why? Sometimes we have such large range, those both numbers is very small, because sometimes scientists cannot replicate all 13 CODIS loci, because sometimes biological evidence can be very degraded, and they would use only 5, 6, 7, 8 loci in order to do their calculation. So if they have all 13 loci present in biological evidence, then numbers can be 1 in billions. But if they would have 5 or 6, numbers can be 1 in millions. So still this, as you see, very reliable method, just like fingerprints, to exclude or prove that some person has the same DNA profile as evidence from the crime scene, and this would be a very strong proof that this is biological evidence belong to this suspect. So the correct answer is answer C. And next question, VNTR, or variable number of tandem repeats alleles, are hypervariable regions of human DNA that differ from each other in Answer A. Location of internal sites recognized by restriction enzymes. Answer B. Variable number of point mutations. Answer C. Number of copies of internally repeated DNA sequence. Answer D. Variable location on different chromosomes. And answer E. Ability to hybridize with specific single locus probe. And the correct answer would be answer C. Number of copies of internally repeat DNA sequence. So, as I said, in CODIS system, and CODIS stands for the Combined DNA Index System that were developed by FBI, we have 13 loci, and in each locus we have different number of tandem repeats of the core sequence. For example, this can be T, A, C, G, and in some people this core sequence can repeat 2 times, in other people it can be repeat 4 times, and scientists made special tables of occurrence of different alleles. So if this core sequence repeat 3 times, this would be one type of allele. If this would repeat 4 times, this is going to be different type of allele for the same locus. So normally one locus has between 5 to 15 different alleles with different number of repeats, and these repeats that can be different in each person flanked with 3' sequence and 5' sequence that is the same in each person. So scientists able to create primers for 5' end and for 3' end, and replicate this sequence between these two primers. And because we have 2 chromosomes in each pair, so we have a total number of 23 pairs of chromosomes. On 12 of them we have 13 sites, so one chromosome has 2 loci, but the rest have 1 loci, not all 23 of them, only 13 of them. And as I said, because we have all chromosomes in pairs, one copy comes from mother side, another from father side, we may have at the same locus on the same pair of chromosome different number of tandem repeats. For example, on the chromosome that we got from the mother side, we may have 3 repeats. In chromosomes that we got from the father side, on the same homologous chromosome, we may have 5 repeats. On the next chromosome, we may have different number of repeats, can be 6 and 8, and of course core sequence can be different, because this is going to be different chromosome, different locus, and different core sequence. So as you see, every person has a unique number of such tandem repeats, and no 2 people have the same DNA profile, just like no 2 people have the same fingerprints. Except, of course, when we talk about DNA profile, 2 identical twins also would have the same DNA profile, but all the other people would have different DNA profiles. And when we have DNA profile for all 13 loci, we can say that probability that 2 people would have the same DNA profile would be like 1 chance in trillion. So as you see, DNA profiling is very reliable method. That's why it's become a standard in forensic science. And this is all for today. Thank you for attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.